Welcome to Biology Made Easy. In this lesson, we are discussing C4 plants. We will discuss examples of C4 plants, the habitats, anatomical characteristics, and adaptive significance of C4 plants. C4 plants includes 5% of all plants. Now, examples, we have sugarcane, corn or maize, millet, sorghum, switch, grass, and these examples you can see they are all monocots. Their habitats in the tropics where there is high temperature, high light intensities, dry conditions. So in such environment, stomata will close during the day to reduce transpiration, to conserve water. And once stomata is closed, carbon dioxide cannot go in, oxygen cannot come out during the day. And C4 plants in such environmental conditions have anatomical characteristics that enables the plant to live and photosynthesize successfully. So the anatomy is more of the leaf. So here is transverse section of a leaf. We have the cuticle, upper epidermis, the palisade, you have spongy cells, and typical of C4 plants, we have two distinct close-fitting cells around the vascular bundle. So this is what I've drawn here. This is a vascular bundle. You have this close-fitting cells around the vascular bundle, and this close-fitting cells is called bundle sheath, and you have an outer close-fitting cells which are mesophyll cells, and they are close-fitting and form a kind of a sheath. So you have an outer ring of close-fitting cells and an inner ring of close-fitting cells. And you see the outer ring of close-fitting cells are exposed to air because atmospheric air comes in here and fills these intercellular spaces, and the mesophyll cells are so much exposed to the atmospheric air. But look at the inner ring, that is the bundle sheath. They are not exposed to air at all, and very close-fitting cells. Now, these two rings of close-fitting cells around the vascular tissue have chloroplasts in them. And the chloroplasts are said to be dimorphic because the chloroplasts in the mesophyll cells have grana, and the chloroplasts in the bundle sheath have no grana, it's agrana as we see. So if you compare the anatomy of the leaf of a C4 plant, it will be very different from a C3 plant. Well, C3 plants can also have bundle sheath with chloroplasts, but not the mesophyll sheath, and the palisade cells are quite close together. The air spaces in the mesophyll layer are not so huge as in the C4 plant leaf anatomy. The next thing we want to discuss is that what is the adaptive significance of C4 plants? Why these two rings of close-fitting cells around the vascular region? The adaptive significance of C4 plants, as we said, during high temperature, high light intensities, and dry conditions, the stomata will close during the day. And you know, during the day, photosynthesis takes place because the light is there. Light dependent reactions will go on, and light independent reactions also go on. But if the stomata are closed, then it means carbon dioxide can't get in. Oxygen that is produced during photosynthesis can't also come out. And such conditions, the oxygen concentration will build up, and when the oxygen concentration builds up, photorespiration will take place. We discussed photorespiration in our last lesson. Photorespiration takes place in these kind of conditions, producing phosphoglycolate and glycolate, which is toxic. So these conditions in C3 plants, it happens, but not in C4 plants. C4 plants, the conditions will be this: high temperature, light conditions, dry conditions, and yet photorespiration will not take place photosynthesis will take place and produce a lot of glucose. And then C4 plants have adaptation to reduce oxygen concentration to prevent photorespiration. And this is how it's done in the bundle sheath. Oxygen concentrations are reduced to prevent photorespiration. Air is cut off completely from the bundle sheath because of its thick, close-fitting cells that will not allow air to pass through. Air will not move from the vascular bundles into the bundle sheath because there's no air in the vascular bundles at all. So no air comes here. So no oxygen is completely reduced or cut off to prevent photorespiration. This one. Then another way to prevent photorespiration in the bundle sheath is the absence of grana in the chloroplast. You know, when grana is available, is there will be photophosphorylation to produce ATP, NADPH, and oxygen. But then there is no grana in the chloroplast of the bundle sheath. So no oxygen is produced from photophosphorylation. That is this point. Then the last point to cut off photorespiration in the bundle sheath is that the bundle sheath cells, they have rubisco. You know, rubisco is ribulose biphosphate carboxylase. It has that enzyme. And rubisco has a very high affinity for oxygen to oxygenate ribulose biphosphate to form phosphoglycolate and glycolate. But even though the bundle sheath have the enzyme rubisco, photorespiration will not take place here because the oxygen concentration is very low here, and that cuts off photorespiration. That is this one. The enzyme rubisco does not have enough oxygen to make ribulose biphosphate oxygenate to form phosphoglycolates by photorespiration. So respiration is prevented in C4 plants in the bundle of sheath. Now, how is photorespiration reduced to the minimum in the mesophyll? The mesophyll cells also are close-fitting. Even though they are exposed to air, we have a lot of air here, the cells will not allow air to diffuse in. Any air that gets in should dissolve in the moisture on the surface of the cell wall. So carbon dioxide dissolves and forms carbonic acid that diffuses into these mesophyll cells. So the mesophyll cells also have a way of reducing the amount of oxygen that comes in because of the close-fitting cells. That is it. Then these mesophyll cells, as we said, has chloroplasts that have grana, and so these mesophyll cells undergo photophosphorylation to produce ATP, NADPH, and oxygen. Good. But even though the oxygen is produced in there, it does not undergo photorespiration because the mesophyll cells do not have much ribulose biphosphate carboxylate, this enzyme. These mesophyll cells rather have the enzyme called PEP carboxylate. PEP means phosphoenol pyruvate. So PEP, phosphoenol pyruvate carboxylase, that's the enzyme in the mesophyll cells, instead of having rubisco. So the mesophyll cells undergoes photophosphorylation to produce ATP and it produce oxygen, but the cells do not have enough rubisco to allow photorespiration to go on. The cell have PEP carboxylase. They have the enzyme PEP carboxylase. And PEP carboxylase does not have much affinity for oxygen. So even though photophosphorylation goes on here because it has grana and it produces oxygen, there is no rubisco to attach the oxygen to ribulose biphosphate to undergo photorespiration to form glycolate. So these are the ways by which the mesophyll cells also reduce photophosphorylation. 1, by not allowing air to get through easily, you have to dissolve. 2, it has grana to undergo photophosphorylation to produce ATP, NADPH, and oxygen. But even though it produces the oxygen, it doesn't have much rubisco to do photorespiration, but it has the enzyme PEP carboxylase that does not have affinity for oxygen. So this is how photorespiration is reduced or cut from the mesophyll cells too. So the C4 plants have adaptation to reduce oxygen concentration and prevent photorespiration. We've explained. The C4 plants also are adapted to increase carbon dioxide concentration and increase photosynthesis by fixing carbon dioxide twice. So carbon dioxide concentration is high in the bundle sheath because carbon dioxide is first of all fixed here into a compound called malate, a C4 compound. And the C4 compound is pushed into the bundle sheath where C3 reactions go on to produce a lot of photosynthesis. So the plants are adapted to increase carbon dioxide for photosynthesis because the carbon dioxide is fixed twice. 1, the first time in the mesophyll cells, carbon dioxide is fixed into a carbon-4 compound, malate. And then the malate is pushed into the bundle sheath and carbon dioxide is fixed again into a 3-carbon compound, glycerol 3-phosphate. And then photosynthesis goes on as C3 cycle. So C4 plants are adapted to do 1 cycle of photosynthesis in 2 cells. If you look at what is happening, 1 cycle of photosynthesis takes place in these 2 cells. Once, so I have drawn these 2 cells here. This is the mesophyll cell. This is the bundle sheath cell. This mesophyll bundle sheath here. And in the mesophyll cells, light-dependent reactions go on here. Light comes in here. Water is broken down, photolysis. Carbon dioxide as carbonic acid diffuses in here. Here there is grana, so photophosphorylation takes place. That's the light-dependent reactions take place here to form ATP, NADPH, and oxygen. Light-dependent reactions takes place in the mesophyll cells, and also carbon fixation also takes place to form malate here. Then in the bundle sheath, no light-dependent reaction take place. In the bundle sheath, only light-independent reactions take place here to form glycerol 3-phosphate. So in the bundle sheath, only C3 cycle goes on here. So you see, in C4 plants, 1 photosynthesis cycle is divided into 2 as it were in the mesophyll cells, light-dependent reaction plus some fixing of carbon dioxide. In the bundle sheath, solely light-independent reactions to form glucose here. In our next lesson, we'll look at the process of C4, the pathway, how substances are formed, and the reactions in our next lesson. The detail of what is happening here. But you see how neat plants in very dry conditions are able to survive and produce a lot of food because of the adaptations that we have talked about here. So we'll meet in our next lesson. If this lesson has been useful, give us your likes and kindly subscribe. Thank you very much. Bye-bye.